It's really my pleasure to introduce Dr. Jason Snape from AstraZeneca, Global Safety, UK. Jason Snape is a senior principal environmental scientist, and his background is on environmental risk assessor. It's a microbiologist and biochemist. And his work is mainly dedicated to the support to support science and evidence-based policy and regulation. And so he's very active in many European bodies, like the Federation of Pharmaceutical Industry Association and the Environmental Risk Assessment Group. So I don't want to take any more time because we have already still a few minutes to the keynote speech. So please, Dr. Snape, the floor is yours. You can navigate into your presentation. I guess you have already been instructed on that. And so please, enjoy us with your presentation.
Okay, thank you very much. It's an absolute honour to be here, and I'd like to thank the Scientific Organising Committee for the invite. The subject I'm going to talk about, obviously, is going to be something that if it's not very close to your heart at the immediate moment, it's probably something that may touch your life in the years ahead. Obviously, I work for AstraZeneca, but I'm also an honorary professor at Newcastle University and at Warwick University. And for the Harry Potter fans amongst you, obviously the last time you might have seen Professor Snape was being mauled by a snake in the boathouse at Hogwarts and shedding tears into a vial so Harry could go and see his thoughts in a Pensieve in Dumbledore's office. But I now actually work for AstraZeneca, not in potions, but actually in environmental science. I don't have my cap and gowns, but I do have my wand. So any difficult questions, then I will strike you down at the end. So back to the serious topic of resistance and managing the risks. I'm going to go on a little bit of a journey, but as I said, it's a journey that will touch most people. And I know it touched me probably about 3 years ago. So my father had prostate cancer, had his prostate removed. My daughter had a burst appendix, and actually both had their operations. They were reasonably successful operations, but actually within a week, they were both back in hospitals for extensive periods of time. They'd actually picked up microbial infections, quite resistant infections, and actually took quite a time for them to actually find a regime that can actually treat both of them. And actually that brings quite a lot of heartache and emotional cost to those close to them, as well as them personally. But actually for the healthcare system, it places a huge burden. So they had to go back into hospital, spend in one instance several weeks in hospital, going through extra care, taking up a bed that somebody else could be using. So obviously it's something that I've seen personally, and unfortunately I think many more in this room, if they've not experienced it already, will see it in the years to come. Obviously, I'm going to talk and take us on a little bit of a journey over the next 45 minutes. Part of it's going to be this increasing concern about resistance. I'm going to talk a little bit about what antibiotics are, their mode of action, the different types of resistance we see, and how resistance can spread between microbial communities. I'm going to talk about how we currently do a risk assessment and how that applies to antibiotics, and look at some of the protection goals that we've got and how good or how bad they are. I can't talk about antibiotics without talking about their drug production and some of the problems in India and China. And then I'm going to just focus a little bit on some of the ongoing industry challenges and societal challenges we see before just moving on to a few take-home messages. The picture I've got here, most of you will actually recognise the old distinguished chap as being Sir Alexander Fleming. So he discovered penicillin back in 1928. So a chance discovery of a mould growing on a contaminated plate of a streptococci that he had when he was just looking at some normal kind of chemical-based treatments with kind of sulfonamides and some other compounds. The lady stood with him is actually the first ever patient to receive penicillin. It's a lady called Anne Miller. So she had a miscarriage back in 1942. and she picked up a serious infection, a streptococcal infection as part of that. They tried surgery, they tried several blood transfusions, they tried sulfonamides, and none of them worked. So she actually received the first dose of penicillin, and it was half the US supply and stock they had to treat her. And within a few days, the temperature had dropped from 107 Fahrenheit, so that's over 41 degrees C, where she had extreme septicemia and was very close to death, to actually being healthy. So that was in 1942, and she died in 1999, so 57 years later. And she was a nurse, so actually she gave back an awful lot. So when we think about drugs in the environment and maybe some of the consequences that they may bring, I think one thing we should never lose sight of is that behind some of these risks that we potentially see, there's always a patient story and a patient benefit. So what about the concerns about antimicrobial resistance? Well, there's been lots of reports within the clinical literature, a lot of data that we've seen in terms of increasing resistance prevalence in E. coli to extended spectrum beta-lactamases. We've seen sexually transmitted diseases such as Neisseria gonorrhea, where actually it's pretty resistant to a large number of macrolides. Now we've seen issues with tuberculosis, MRSA, so methicillin-resistant Staphylococcus aureus, C. difficile over a number of years. And in 2012, the Chief Medical Officer for the UK actually raised these concerns in one of her annual reports. And the little circled kind of document in the middle is just the preface to this report. And actually it just really recognises that actually if there's no suitable antibiotics to treat infection, minor surgery and routine operations could become a thing of the past in the not too distant future. And obviously that got picked up. Within a couple of years, it actually entered the UK risk register, and it ranks alongside things like climate change and global terrorism in terms of some of the threats that are brought to society. And obviously that report led to quite a lot of action, not just within the UK, but much more widely. And the UK government commissioned a series of reports. So we took an economist, Sir Jim O'Neill. He developed a task force. They looked globally in terms of the experts that they reached out to. They generated a series of 9 reports in all, actually looking at what the impact could be on society, what the environmental role, the agrochemical role would be, where the innovation was needed, what the costs, what the benefits were around AMR. And they say this was all as a result of kind of some of Dame Sally's activities. She oversaw quite a significant part of this work. And actually she was one of the early people to recognise the importance of the environmental dimension of AMR. And I know as part of that consultation, this group actually brought in some environmental scientists within this room, some other leading experts. I know I had to meet with her team, and they came to meet some of the staff within AstraZeneca to see the work that we were doing. But in terms of what the costs were coming out of these reports, they were estimating that by 2050, that infectious resistance disease would be the leading cause of mortality globally. So 10 million deaths per annum. So that's basically a death every 3 seconds, and a cumulative cost to society between now and 2050 of about $100 trillion. Obviously, Dame Sally didn't stop there. She actually took some of these concerns further. So she actually moved on to the United Nations, and actually AMR went onto the agenda at the General Assembly meeting in September 2016. And a number of companies have made a series of declarations there. Countries made declarations there, as well as industry alignment to that meeting. And I'll come back to the industry declarations a little bit later on. But the concerns don't just stop at that kind of level. They actually start getting picked up by some of the investor kind of community. And some of this will be around the issue of antimicrobial resistance itself, but also issues around some of the production-related issues going on in China, where there have been high levels of pollution associated with some suppliers to the local market, but also some of the suppliers to kind of the Western kind of markets in Europe and North America as well. So they've come under increased scrutiny. And some investors, more than others, have actually been asking specific questions. So Nordea is the biggest investment bank actually in Northwest Europe, and they're making challenges on industry and the wider society in terms of some of the pollution that we're seeing within India with antibiotics in the environment. They're actually funding research and monitoring programs actually out within some of these countries and some of these regions. In my role within AstraZeneca as kind of the environment director or the senior principal environmental scientist, I actually get pulled in to quite a lot of the corporate social responsibility meetings or the environmental and social risk meetings we have with our investor community. And there were two that were hosted last year, one by Nordea in Stockholm and one by Goldman Sachs in London. So I was brought in and we were put in different rooms with, in both occasions, probably about a dozen different institutional investors. And most of those investors can be anything from kind of capital companies, finance companies, big banks, but they also cover a large number of insurance companies, things like Standard Life, Legal in General. And they ask quite critical questions. And most people in the room, maybe not the students or the PhD students, but most people in the room will maybe have a life insurance policy or they will have a pension kind of, which will be with some of these companies that we're asking some of these questions. And obviously they invest in companies such as ours. So indirectly, if you have a life insurance policy or you have a pension, you're probably an indirect shareholder in one of the major pharmaceutical companies that be represented in this room or further beyond. And they're now starting to ask questions of us. So out of 12 of the investors in the Goldman Sachs room that I was in, probably 10 of them were asking us, what are you doing to manage the issue of antimicrobial resistance? What are you doing to manage your supply chain and its footprint? And these were all not just questions about drug discovery, drug development, it was about product stewardship and environmental stewardship, about pharmaceuticals in the environment, because resistance is a material risk to their business. They're ensuring life. And actually, if resistance infectious disease meets the levels that O'Neill has predicted, actually they're going to have to pay out on a lot of policies, or people taking out new insurance cover are going to see a lot more exclusions maybe come in some of those insurance policies as we go ahead. So it's a challenge across all levels of society. So we don't only face questions from the scientific community, we get these questions actually from the investor community, and they're on a par with what your drug development and drug discovery pipeline look like. They know what the risks are with that. It's the wider risks that they're starting to look at. We also have kind of sustainability indices like the Access to Medicines initiative starting to include benchmarks on AMR. And that's not just looking at research and development, but it's also looking at manufacturing and production practices, and it's looking at appropriate access and stewardship. And they include things like environmental risk management and disclosure on environmental risk management. So setting protection goals, being visible and transparent with those protection goals to make sure we're managing the risks within our portfolio and within our production. And 30 companies were invited to partake in this AMR kind of benchmark that Access to Medicines conducted. 10 big industry, so research-based industry, 10 generic, and 10 small kind of startup or biotech companies. And they say it's resistance driving a lot of these questions. But it's not a new phenomenon. Resistance has been known since Fleming's time. So back in the New York Times, there's a quote included in this slide where he's saying the thoughtless person playing with penicillin treatment is morally responsible for the death of man who succumbs to infection with penicillin-resistant organisms. So he foretold this even at the time of his own discovery, just as its use was kind of expanding during kind of the Second World War. But more recently, kind of science again has shown it's not a new phenomenon either. So work from kind of Jerry Wright's group back in 2012, published in Nature, has actually looked at permafrost kind of cores up kind of on the Alaskan Peninsula, and in sediment samples or core samples more than 30,000 years old, you're finding highly diverse collections of genes encoding resistance to beta-lactam, tetracycline, and glycopeptide antibiotics. And this is largely because most of the antibiotics we have in the clinic, not all of them, but most of them originate from natural origins. So they're part of the biological or the biochemical warfare that's going on between microbes within nature. And they serve ecosystem functions for microbes communicating between each other with quorum sensing and other activities. and resistance is going to be natural within some of those communities. It's just the industrial scale use that we've got has developed this natural or innate levels of resistance that we found in the environment to something that has gone beyond that, by which it's now transferred from maybe typically environmental organisms in the past to those of clinical or pathogenic relevance. When we're thinking about antimicrobial resistance and antibiotics, I thought it's probably best maybe to just throw a few definitions in. An antimicrobial is quite a broad definition, so it's anything that can kill or prevent microbial growth. And it's not just antibiotics, it includes antifungals, antivirals, biocides, disinfectants, antiseptics. An antibiotic is more specifically targeted towards an antibacterial, so it's a specific type of antimicrobial. And either they can kill bacteria, so they can be bactericidal, but actually they can also inhibit bacteria, they can be bacteriostatic. And as I said, most modern antibiotics are either natural or semi-synthetic derivatives of natural compounds. When we hear antimicrobial resistance, it could be resistance against bacteria, fungi, viruses, or anything else, but it's more synonymous with antibacterial resistance, so resistance to antibiotics. And likewise, antimicrobial resistance genes, or ARGs, are the genetic determinants that actually encode for resistance. And they can be chromosomal based, so actually within the natural DNA of the organism, or they can be plasmid based. And these plasmids can be actually transmitted between bacteria, as I'll come back to a little bit later on. And when we hear the word resistance kind of thrown around, it means a lot of different things to different people. So resistance maybe to somebody in the clinic will be the concentration at which you kind of get clinical breakthrough of a resistant isolant. But when we hear resistance from maybe an environmental perspective, we're just talking about the level of resistant bacteria or the level of resistance genes that are in the environment, irrespective of any maybe adverse ecological or clinical kind of consequence. So we have to be careful sometimes when we're using the word resistance, because it can mean different things to different people. There's lots of different classes of antibiotics. So this is a discovery time scale that we have on. It's not the time scale at which they hit the clinic, because whilst Fleming kind of discovered penicillin before the sulfonamides hit the clinic, actually the penicillins actually weren't in clinical use until kind of maybe 1942 there. But there's lots of different classes. As I said, most of them are natural or semi-natural of origin. There are some synthetic classes within there, particularly kind of things like the fluoroquinolones. They have lots of different modes of action, lots of different structures within there. Some of them are bactericidal, so they kill bacteria, so they're the ones in the dark green. And some of them are bacteriostatic, so they're just present in inhibitory concentrations, which are in the light green. So there are a whole diversity of moieties that we can use, and there's a whole group of different structures within each of those classes. And they have a whole variety, as I mentioned, of mechanisms of action. So on the left-hand side of the image, you will actually see that we've got agents that can inhibit DNA synthesis. We have agents that can inhibit protein synthesis. And we'll have agents that actually act against the integrity or the formation of the cell wall or the cell membrane structure. I think one thing to draw out of this slide is obviously the antibiotics are specifically challenged or targeted towards bacteria. So these are prokaryotes. So these are quite simple kind of organisms in comparison to most of the eukaryotes that the ecotoxicologists will work with within the audience here. On the right-hand side of the image, obviously we've got the mechanisms of resistance. And this can be kind of stopping the kind of incoming or the antibiotic from being taken up into the bacterium. So it's kind of increasing the impermeability of the cell. We can have efflux pumps to actually get the concentration out within the cells itself. The bacteria are smart. They can modify their targets to actually mop up the antibiotics, so stop it hitting the target of action, or they can alter their target. But you can also get things like inactivating enzymes, so beta-lactamases, which we heard quite a bit about in the AMR session earlier today, and also aminoglycoside modifying enzymes. And there are other modifying or inactivating enzymes that we have for macrolide antibiotics as well. The interesting thing with the inactivating enzymes is quite often our industry gets challenged with discovering biodegradable, kind of benign pharmaceuticals. Well, in the instance of kind of maybe beta-lactam antibiotics, they are quite biodegradable antibiotics. The trouble we have here is the beta-lactamase enzyme that biodegrades them, whilst it's good and serving an ecosystem function or service when it's in an environmentally important bacterium, it actually is the cause of resistance and maybe an adverse clinical outcome if it gets into a pathogenic organism. So what's good for the environment isn't always good for the patient in this instance. As I mentioned, resistance can be extremely mobile within microbial communities. So it's not too long ago that it was thought that kind of bacteria reproduce kind of asexually, and you just have one parent cell go to two daughter cells and you'll just get exponential reproduction down that line. Well, that's not the case. Bacteria have sex, and bacteria are pretty good at having sex. They can have conjugation, where they can actually share plasmids through direct kind of intimate contact. You can have transformation, where they can take up naked DNA that's been released by dead bacteria. You can have transduction, so like ourselves, we can get infected by viruses. Bacteria get infected by viruses too. They're called phage, and that can transfer DNA from one bacterium to another. Then obviously alongside that, we can get mutations as well. So they could be chemically induced mutations or sunlight-induced mutations that can, again, convey resistance. So what does an exposure map look like for an antibiotic, if we try to think about it in a little bit more detail? And this comes from a paper that was published just actually earlier this month, led by kind of Joachim Larsen from Gothenburg University, that was a result of a joint program initiative AMR meeting that was held in Gothenburg late last year. And obviously the main routes of antibiotics to the environment and potentially resistance development in the environment come from the use of antibiotics in human populations, their drug production, animal husbandry and animal agriculture, but also aquaculture. And once those antibiotics are actually in use, you can get selection for resistance genes actually within the animal model species, within aquaculture, within the human. So they will be discharging antibiotic residues, but also resistant bacteria, resistant genes to the environment, where you can get dissemination of those genes, you can get selection for more novel resistance genes, and you can get transfer between environmentally important bacteria and pathogenic bacteria. So the environment can act as a source for selection, maintenance, but also evolution of antibiotic resistance. Quite often we hear about, well, what about the classes that are used in human and veterinary medicines? And this is a slide that I've taken with his permission from Thomas Heberer that was presented at the Pharmaceutical Interest Group meeting we had on AMR on Sunday, which just kind of illustrates that whilst we have some human medicinal antibiotics that are exclusively used in humans, we don't have any antibiotics in Europe that are exclusively used in veterinary medicine, in animals. What we do have are some antibiotics that are used in both human and veterinary medicine at the moment. So sometimes when we hear this call of don't use kind of veterinary medicines within human medicine, actually that's not appropriate, and it's not possible. We actually need effective antibiotics actually for veterinary medicine as much as we do for human medicine. Again, this is just looking at exposure, like I had before, in a little bit more detail, and it's just trying to, and this comes from a meeting that was hosted in Montebello back in 2012 that Ed Top in the audience kind of chaired, and I was on the co-organising group and one of the co-funders for, where we were looking at the environmental dimension of AMR really for the first time in a lot of detail. And this was just from a paper looking at trying to integrate human and environmental risk assessment for the first time. And what we could see here is that the inputs to the environment, whether or not they were coming from aquaculture, whether or not they were coming from kind of human discharges and wastewater treatment plants, or whether or not they were coming as a result of farming, manure and slurries, you have resistance genes enter the environment, you have antibiotics entering the environment, but actually you also have things called co-selecting agents. So it's not just antibiotics that select for resistance. We also know things like biocides co-select for resistance, but also metals as well. So actually quite often when you're thinking about risk management strategies where you focus purely on the antibiotic, if you don't consider some of the other co-selecting factors, actually you may not get a reduction in resistance. And obviously when you've got release of any of these genes, antibiotics or co-selective agents, obviously you can actually have uptake of the genes, as I've said before, by transformation into bacteria to make them resistant in the environment. But also you can have the antibiotic and the co-selective agents actually acting as a selection pressure for the maintenance or evolution of resistance in the environment. And that could be in environmentally important bacteria serving ecosystem service functions, or actually it can be in pathogenic bacteria. And within those communities, you can get a lot of horizontal gene transfer, mutations going on, and whether or not that's happening just between environmental bacteria or moving into pathogenic bacteria is still under study. What we know at the moment is probably not quantitative in any shape or form. So I think where a lot of research is actually needed at the moment is actually looking at the relationship between the environment and the clinic. Where a lot of the research is going on at the moment is looking at the dissemination from maybe the farm or the sewage treatment plant and the industry out to the environment. It's not maybe looking back quite as much or actually thinking in terms of where the size of the arrows are in terms of the significant routes actually back to man. Next kind of couple of minutes, I'm going to talk about risk assessment and how that applies to antibiotics and resistance. So when we have a new human medicinal product and this is the guidance we have for human medicinal products, but there's slightly different guidance for veterinary products, we do have to do a risk assessment. That came into force in 2006. Unfortunately, most antibiotics and antibiotic classes were all registered prior to kind of that time, so the amount of data we have is somewhat limited, but I'll have a look at the data we do have a little bit later. So when we're doing a risk assessment, we look at the maximum daily dose, which is extremely high for most antibiotics. We look at the market penetration. We assume we're going to treat 1% of the population as a default, the amount of wastewater treatment, and dilution in the environment. If we have a surface water predicted environmental concentration greater than 10 nanograms per litre, we have to do a risk assessment. All antibiotics would trigger the requirement for that risk assessment because typically they're drugs that we're taking in the high milligram to gram per litre range per day. For that risk assessment, we have to do our phys-chem data, we have to do our fate and behaviour studies, but we also have to do a range of ecotoxicology studies, so activated sludge respiration inhibition tests, algal growth inhibition studies, chronic Daphnis studies, and fish studies. And depending on the nature of those studies, we may have to do a terrestrial or sediment assessment or a bioaccumulation assessment. When we're working on antibiotics and we think about protection goals for antibiotics, we wouldn't do an algal growth inhibition. We'd actually do one with cyanobacteria because obviously our compounds are targeted towards treating bacteria. So if we have a look at in the public domain for all the NOx and the EC50s that we can find for cyanobacteria, Daphnia, and fish, what does that tell us? And this is some work that my company co-funded with the University of Exeter that's ongoing in a paper that was published late last year, is we've looked at the NOx, and this is a graph of NOx in the first instance, to actually look at the log sensitivity across a number of different tests. So the horizontal dark line that we have there is the normalization point for cyanobacteria. So that's what we think is the most sensitive species within our ERA. And what you can see is if we look at clinically relevant bacteria, they're probably as sensitive on a generic basis as the cyanobacteria. And they should be because they have those drug targets in the most part. If we look at Vibrio fischeri, some microtox, again, that's a microorganism. Its median sensitivity is probably a little bit more sensitive than the cyanobacteria. If we look at the activated sludge community, so the wastewater treatment community, where we can't characterize the innate resistance of that community, it's not particularly sensitive. And this is a log scale that we have on the y-axis. Again, if we look at the macroalgae, in the most part, they're not sensitive. The same with macrophytes, invertebrates, and fish. The one dot that we have for fish that actually is more sensitive is because the company tested a prodrug and actually not the active ingredient. If they tested the active ingredient, it would have fallen above the line. So that raises the question that actually, at the moment, most of the species we test actually provide nothing in terms of protection goals towards risk assessment other than the cyanobacteria, because we don't use Vibrio fischeri, we don't use the clinically relevant data, but actually all the data we have on invertebrates and fish doesn't drive our risk assessment. Now, the slight issue we have is we only routinely test one cyanobacterial species. So we test Anabina flossaqua, and that's indicated by the little arrow and the blue circle on this diagram for a range of antibiotics. This is published data. And again, it's a log scale on the y-axis. So what we're seeing in this instance is that for some antibiotics, other species of cyanobacteria that obviously serve a primary productivity function in nature can be up to 4 times more sensitive than the species that typically used within the regulatory risk assessment. So we have some question marks actually over our current approach to the risk assessment of antibiotics. And there's a poster that's out there currently at the moment by Gareth LePage actually looking at that species variability in a little bit more detail. We think more widely in terms of the protection goals for antibiotics that have been advocated. Joachim Larsson's group in Gothenburg advocate the use of clinically relevant data. So if we have a new antibiotic, quite often when it's approved for several years after it's been approved in several countries, we have to look at the clinical efficacy. So if somebody presents in a clinic with an infectious disease, the sample will get taken from that patient, and we will see whether or not it's resistant to the organism, to see whether or not the efficacy of that substance or that antibiotic is being eroded. Likewise, if anybody presents in not just for that antibiotic into one of the labs and they're being tested, a lot of that data will get collated, and they will see what the minimum inhibition concentration is for that drug against that pathogen that's been isolated. And that data gets collated in the EU-CAS database. So it's making sure that you're still treating the patient at an efficacious level with that drug, or you're still treating the patient with a drug that still works. So Larsson actually took this data, he looked at the lowest 1% for the range of species that were identified, corrected against the number of species, and he came up with 111 protection goals that he called a PNEC for resistance, and it's a theoretical value. And you can see that covers a huge range of kind of sensitivities from 8 nanograms per litre up to 64 micrograms per litre. So we compared those protection goals from the LePage kind of work, which has our normal kind of environmental kind of ecotox data, with the data that came out of the Benson-Palm paper. And what you can see actually on this graph is that for compounds where we had ecotox data, for which there was the MIC-derived data, if it's above the line, then actually the environment is driving the protection goal. If it's below the line, then the clinical species are driving the protection goal. So actually, when you're thinking about protection, you need to think about maybe some of the clinical isolates as well as the environmental isolates. And we don't currently do that within our approach. Within the AMR Alliance, which I'll talk a little bit about later on, we've actually shared a lot more data since the LePage paper has come out. And actually this relationship holds true actually for a bigger number of compounds at the moment. So industry, when it's looking at protection goals at the moment, is looking to apply the lower of the two values within its kind of effluent management programs. That's what it's advocating at the moment. I think the other thing to consider is where we're looking on our dose response curve as well. So obviously a lot of the environmental kind of endpoints are derived from kind of EC10s or NOx or EC50s, but actually the MIC is actually the equivalent of an EC100. So an MIC is the minimum inhibition concentration, so that's the lowest concentration at which you get complete inhibition of the pathogen. So it's designed to tell the doctor what concentration he needs to be giving to the patient, not what concentration needs to be protecting the environment. And when you're deriving an MIC, you're not looking at the full dose response curve. But they're not the only protection goals that potentially exist for resistance and antibiotics. We have the tools to go out and measure resistance genes. Should we be treating genetic determinants for resistance as pollutants? there. They have the ability to get into other environmentally important bacteria and to pathogenic bacteria. But the trouble is we don't know what levels are safe. We don't know what the routes are back to the environment. We don't know all the resistance mechanisms. And this assumes that we have a priori knowledge of the resistance genes in order to be able to go and track them. There are a range of activity-based assessments that exist to look at antibiotic residues in milk that are approved by the FDA. However, these have low sensitivity, normally at about 50 micrograms per litre. But we've already seen actually the environmental kind of sensitivity can be quite a bit lower than some of those kind of residue kind of tests, although the benefit for those is they don't only look at the antibiotic, they will look at any transformation products that also have any antibacterial activity. We're seeing some groups advocate looking not just at the number of resistant E. coli in surface waters or in sewage effluents, which we would use for kind of things like the bathing water standards, but it's extending that to look at the number of resistant E. coli. So things like kind of ceftaxime, which is a cephalosporin, so a beta-lactam antibiotic. And that would work particularly quite well maybe for domestic sewage treatment works, where there's obviously kind of a fecal input and enteric input to those plants, but actually it wouldn't work well for industrial manufacturing plants, where obviously there's little or no fecal input. And then there's a whole area of science coming through, and the papers illustrate that on the right, and we had 2 presentations this afternoon in the AMR session, looking at minimum selective concentrations. So what are the environmental concentrations that select for resistance? But some of these methods need validation before they can be used in environmental risk assessment. So I'm going to just talk a little bit now from the data that we do know, both from the Larson paper and from the LePage paper, about what can we actually do in terms of environmental risk assessment. So this slide, I'm just doing some work looking at environmental exposure assessments. So I've done a worst case PEC, so a worst case predicted environmental concentration for each antibiotic where there's EU kind of coverage or EU use. And I can get that consumption data from IMS Health. And we've got 91 antibiotics in which we can actually look at that, and they're captured on the right-hand side. And anything that's red has a median exposure concentration above 100 nanograms per litre. Anything that's green, its concentration will be below 100 nanograms per litre. And the width of the red or the green bars just shows the variation in use across the EU countries. And this is worst case, so it's assuming 100% patient use, no metabolism, no degradation of that compound. We can take that data that we've got on PECs and then actually do a risk assessment, so we can actually look at the PNEC that Larson and Benson, Palm and Larson have for resistance, but also the environmental limits derived from kind of the ecotox data, and actually look at a risk quotient for each country and each of the APIs. And again, I've color-coded these just as kind of high risk, which is red, which has a risk quotient of 10, orange, which has a risk quotient between 1 and 10, yellow, which has a risk quotient of 0.1 and 1, so low risk, and then green has a risk quotient of less than 0.1 as being insignificant. So the left-hand graph is just showing the risk in terms of the ecotox data that we have. So there's only a limited number of compounds for which we have ecotox data in the public domain. But again, that spans about 7 orders of magnitude in terms of risk quotient across the bottom. On the right-hand side, we've used the Larson MIC derived kind of risk quotients. And typically what we're seeing here is there's one beta-lactam that's kind of has a risk quotient kind of greater than 10, actually based on the ecotox data. But this is assuming no degradation, no removal. The interesting thing with this is I could factor in biodegradation of that compound, so that amoxicillin compound, quite easily. And its biodegradation would be extensive, so it would actually remove the environmental risk of that compound back down to green. Unfortunately, that the enzyme that actually degrades it, if it gets into a pathogenic organism, could kill people. So in revising the environmental risk down, I'm potentially revising the human risk actually up. But in most instances, what we can see if we're looking at the MIC-derived data, if that's protective, is that most antibiotics don't pose a risk on worst-case assumptions at the moment. But this is around patient use, not drug production, which is what I'll move on to at the moment. So what about drug production itself? Well, Larson back in 2007 and his co-workers found up to 31 milligrams per litre of some fluoroquinolones in an industrial kind of effluent site within Hyderabad region of India. And that's not isolated. He did a review in 2014 looking at a whole range of kind of production sites across a range of countries. And again, there were some high levels associated with antibiotic and other drug production in some of these kind of regions of the world. And that recognises there is no regulation that determines what levels drugs can be discharged to, either within Europe or anywhere kind of globally. And that raised some questions. And it's obvious that it's unacceptable to have discharges at 31 milligrams per litre going in to the environment. And Larson's done some calculations that based on the loads and the volumes flowing through that plant, that 31 milligrams per litre corresponded to what Sweden would typically prescribe and use within a week. So it's high levels that we're entering the environment. So industry, as part of kind of Dame Sally's effort to bring AMR to the UN, actually responded with a roadmap. And within that roadmap, its first commitment was around reducing the environmental impact from the production of antibiotics. So that's one of the key messages that we put across, alongside things about new R&D within that area, around things about responsible kind of use, diagnosis, prescription, and use. And in more detail, if we look at the signatories to that response, so there are 13 companies that actually signed the UN roadmap kind of response out of 100 companies that are actually within the AMR Alliance. We committed to review our own manufacturing and supply chains. That's ongoing, largely complete at the moment, so we know where there's good and bad practice. We've published a common framework for managing our antibiotic discharges, as our commitment was by 2018. We're still working on a practical mechanism for transparency, but we have been sharing data, and we're moving towards kind of having an approach for discharge concentrations, and that's something that Joan Tell from Merck shared with the FIG group and sought feedback actually on Sunday this week. And kind of our proposal is to apply the lower of the traditional environmental PNEC and the Benson Palm PNEC resistance. If you want more information, the AMR Alliance issued its first report in January this year, has run a number of case studies. I know one of the case studies was the LePage et al. Work that I referenced earlier on. But in addition to looking just at that environmental component, antibiotics do pose a bit of a challenge in drug discovery and drug development as well. And that's something else that the AMR Alliance and other stakeholders are looking at. So when we do drug discovery and drug development, it's estimated that it costs about $2.6 billion to get a drug to market. And that's factoring in attrition as well in those drugs that don't make it. So we start off with thousands, tens of thousands of candidates in drug discovery and preclinical kind of investments and investigations. We move into our phase 1, phase 2, phase 3 clinical trials where the number of drugs rapidly decreases to get to one medicine that will actually go through for regulatory kind of submission and FDA review. And it's somewhere in the region of about 12% of the candidates that actually move in to phase 1 clinical trials actually ever get approved. So we're a high-risk business. We have a fixed patent life on these new medicinal products. It's 20 years. It's not 20 years from the point of FDA review and approval. It's 20 years from the point of drug discovery. So the amount of time that we have in development and clinical trials is eroding our patent clock. And typically, it takes 10 years to get a medicine to the market. So we have a 10-year payback time on any new medicinal product coming through. What does that mean for antibiotics if we're looking at the investments, looking at the economics there? Well, actually, it takes 23 years to get the return on your investment for a typical antibiotic. So this came from Lord O'Neill's report. Our patent life runs out in 20 years, so there is no return on investment currently for antibiotic discovery. If we have resistance breakthrough 5 years after launch to market, you make even more of a loss on it because it's eroded that return on investment that you've got. We've also got an economic model at the moment where sales are linked to volumes, which is not what you want if increased sales are going to result in higher levels of resistance. If you come up with a new class of antibiotic, it's more likely to be kept safe and locked away in a cupboard as a last line of resort, yet it's got a ticking patent clock and is going to lose exclusivity and payback. So you need a new model. So a lot of the work that's going on between government and industry and a variety of other stakeholders at the moment is to look at new models for drug discovery in this area. And a lot of that is resulting in a shifting model. This was quite a scary kind of fact that I read from a report from Chemistry World in March this year, where basically in the UK at the moment, it's estimated there's probably about 110 industry scientists in big business working in the field of antibiotic discovery at the moment. I know in the UK there's more than 110 environmental researchers tracking the issue of antimicrobial resistance in the environment. And if you draw contrast to cancer, we've just got a single kind of charity in the UK, Cancer Research UK, supporting about 4,000 researchers. So actually it's an under-invested area, and it's not quite as bad as it seems within this slide, because actually what we're seeing now is a lot more public-private partnerships. So we got things like CARB-X, which is a partnership between the US health departments, the Wellcome Trust, which is throwing kind of almost $1 billion at drug discovery and drug development. But you've also got partnerships between the European Commission and FPA, so the European Federation of Pharmaceutical Industry Associations, through the IMI program, the Innovative Medicines program, again investing somewhere in the region of about 700 million euros at drug discovery. So the nature's changed from in-house big business discovery to kind of shared cost, shared responsibility, shared discovery and shared benefits between the public sector and the private sector. So I'm just now going to move on just to a few take-home messages. Kind of conscious we're pretty much at time. One thing we shouldn't lose sight of is resistance provides an ecosystem service. It's a cruel fact, it's true, but actually it's adaptive, it's protective of a lot of microbiological habitats. The one thing that we need to make sure of is it's not compromising resilience of some of these kind of ecosystems itself. If you reduce antibiotic concentrations in the environment, we can reduce the selective pressure for AMR evolution and the development and maintenance of resistance in the environment. But we have some issues with our current approach to chemicals management. The chemical standards that we currently apply, maybe at the end of the pipe, don't always work for antibiotics and resistance. So they're not going to address things like antibiotics leaving the patient before the wastewater treatment plant, where there may or may not be selection occurring. These chemical standards don't look at resistance genes or resistant bacteria leaving the patient or the wastewater treatment plant, or the resistance genes being enriched within that wastewater treatment plant. And none of them are looking at the presence of co-selective agents in influents and treated effluents. We also have this issue where high concentrations of an antibiotic don't always equate to high concentrations of resistance. So if the resistance mechanism is degradative on there, i.e. the enzyme that encodes for resistance biodegrades the antibiotic, where you've got low concentrations could correspond to where there's high concentrations or high levels of resistance. From an ERA perspective, they don't cover resistance. At the moment, we look at the impact of the antibiotic, but the data that's coming out at the moment tends to indicate, particularly some of the MSC data that's coming out, the minimum selective concentration data that's coming out, is the ecotox data may be sufficiently protective in many cases, not all cases, but at least it will lower the protection or lower the potential for selection and evolution. Our current ERA process is too reliant on a single species of cyanobacteria at the moment, and that cyanobacteria we've seen is highly variable. So we need to bring in more ecosystem services provided by bacteria actually into our risk assessment processes. And that's a conclusion advocated by Christian Brandt within his paper in 2016 and supported by the LePage paper in 2017. And we advocate using a variety of cyanobacteria within that risk assessment. And obviously we've got this issue of co-selection with other chemicals. So a pure focus on antibiotics might not manage all our risks. But again, we've got to think wider than the environment itself, that actually when we're looking at some of the roadmaps within Europe and within some of our countries and by the World Health Organization, that actually responsible prescription, responsible diagnosis and responsible use could actually have a greater reduction on the burden of AMR than actually any other environmental intervention. It's anticipated within the O'Neill report that up to 30% of prescriptions of antibiotics are against infections that would never respond to it. And in this instance, on the right-hand side in the image, we're looking at the 40 million people who get antibiotics to treat respiratory issues. Only 13 million of those patients actually needed those drugs. 27 million actually didn't. So responsible use, diagnosis and prescription can lower the environmental burden from patient use, but also lower the burden from manufacturing and production. So my final kind of slide is just that a reflection that most clinical surveillance programs that we have within our public health laboratories at the moment do not include environmental matrices whatsoever. But what we are tending to see is sometimes that the environment may be a canary or an early warning kind of system to what might happen or what might occur within the clinic. We're seeing novel sulfonamide, novel quinolone, novel carbapenem resistance mechanisms in the environment that we're not seeing within the clinic at the moment. So is the environment a smoking gun to what we may see in the clinic in the future? What is essential is a One Health approach. We need to think about animal health. We need to think about the ecosystems and environmental health. We need to think about human health and actually work together to tackle this issue. And it's not an issue that can be tackled by one country or one stakeholder. It is truly a global issue. And it's the kind of issue that SETAC could help foster and bring together to solve. And then the final kind of reflection that I have within this slide is, at the moment, if I'm sourcing the production of drugs throughout the world, my company will actually have quite tight environmental criteria on where we source our drugs from. So who our suppliers are, who they're formulating them. We give them safe discharge concentrations. We audit compliance with those safe discharge concentrations. But the people buying those drugs from my company don't ask those questions of me at the moment. So the people that are providing the medicines to you, the governments that are buying those drugs, the healthcare providers. In many cases, they will actually buy the cheapest generic version that they possibly can. So you could ask that challenge, that if they're having that auction on a monthly basis, actually in buying the cheapest, are they actually buying some of the pollution problems that we're seeing within India and China? So that begs in turn a question that will society actually pay for responsibly sourced antibiotics? And given the threat of resistance that we face with the numbers that have been projected by Lord O'Neill, can we as a society afford not to? And I'll just end with just some acknowledgements to some of my co-workers at the University of Exeter, and I'll thank you for your time and attention. Thank you.
Thank you very much. I think we have time for some quick questions. I think there will be some of the public.
There's one over there on the right.
Oh, yes. Please state your name, please.
Okay, I'm Clara Hilshova from SETAC, Czech Republic. Thank you very much for this very, very interesting talk. It's actually very interesting and also very raising a high concern. And I was wondering, in this environmental risk assessment, also in the potential assessment of risk for resistance development, are actually joint actions and mixtures of antibiotics taken into account in some way, because you have shown some risk of the individual ones, but they are co-occurring in the wastewaters.
So the question was about kind of environmental mixtures being kind of brought into account, and currently the environmental risk assessment process that we follow is on a substance by substance basis, but obviously resistance here it tends to be quite class-wide in many cases for some of the mechanisms that we're seeing. So actually, I know some of my students are now actually looking at mixtures in terms of some of the work that they're doing, but from a regulatory perspective, it's not included at the moment. But we also know that as well as some resistance mechanisms kind of being class-wide, a lot of the bacteria are resistant to a whole variety of classes as well. So again, that complexity is something that probably needs to be brought into some of the scientific studies that we need to look at actually moving ahead, and to determine kind of that selective and evolutionary pressure that kind of that mixture will actually pose, rather than looking just on a compound by compound basis. But yeah, I concur.
Henrik Thyle from the Danish EPA. How much does the current veterinary use of antimicrobials, and even maybe non-veterinarian use, but used in agriculture, constitute to the problem? And if it is a problem or will be a problem, do we have any ideas on how to control it?
I think in terms of the relative contribution of kind of veterinary versus human use to kind of the problem of AMR is one that still needs kind of careful resolution. There's been a lot of interventions within kind of some kind of regions to kind of tackle kind of the issues with veterinary use. So obviously the ban within Europe in 2006 on growth promotion is kind of a positive step, but obviously there are still regions within the world, so I think it's 33 countries, where kind of growth promotion of up to 39 antibiotics is still allowed. We still, from a human kind of therapeutic kind of use perspective, have over-the-counter use in many countries, so that still needs to be tackled. So I think some of those might be some of the quick wins that we've got, is in terms of kind of responsible use, responsible diagnosis at the moment, and actually tackling this in terms of global stewardship, because we can get our house in order in Europe over the years ahead, but unless India, China and some of the other regions within the world follow, given the nature of global transport and kind of the global food chain at the moment, that might not be enough.
Are there any other question? Oh, yeah, there are two. Please, quick question.
I was thinking a chemical risk assessment at... Oh, sorry, I'm Rick Oldenkamp from Radboud University. Chemical risk assessment, as we do it, is basically based on toxicity and a direct relation between exposure and effect, higher exposure leading to higher effect, and so on. And based on that, we derive PNACs and PEC-PNAC ratios. And I'm not sure whether the issue of resistance, which is not a toxicity thing, but rather an evolutionary adaptation over time and history thing, whether that fits with using, say, threshold values in a more traditional risk assessment way, and whether there might be other ways to look at the problem.
Yeah, I probably concur in some areas. Obviously, the risk assessment we have to do to judge the impact of the antibiotic actually on some of the key critical ecosystem services, essentially kind of primary productivity, maybe nitrogen kind of cycling and things like that at the moment. In terms of the resistance element itself, that's a much harder one to actually bring in, because actually resistance can pose an environmental threat indirectly if it kind of impacts resilience at that kind of community structure level with some of these microbial communities. But actually, at the end of the day, the protection goal in this instance is kind of animal health and human health. And then it's really understanding the relationships of how we can actually bring environmental exposure and selection in the environment actually into some of those human health assessments. But I think one of the starting points that probably we can do is looking at that relationship between minimum selective concentrations and our traditional kind of ecotox protection goals, because if we can minimize the level of selection and enrichment and evolution of novel resistance genes in the natural environment through patient use or through drug production or veterinary use to a level that is relatively safe, then actually that potential human risk will be kind of mitigated to some extent. But that's assuming that the environment is a significant reservoir for kind of clinical infectious disease.
Christian Brandt from University of Copenhagen. Antibiotics are often used together with metals like copper and zinc, for instance, in pig production, aquaculture production, and so on. Hence, one might hypothesize that you get multi-drug resistance development, and you will see maybe even increased co-selection in the future because of the assembly of new mobile genetic elements containing multiple resistance genes, for instance. So how do you think, should we, when we do risk assessment, for instance, of metals and biocides, should we take this into account when we do that? Do you have any opinion on that?
For me, I think that co-selective issue is something that needs a lot more research kind of investigation in terms of the mechanisms for that. There's no great surprise that co-selection or co-regulation of some of these resistance genes exists because, as you said, within pig farms, you've got that common use of kind of metals, biocides, disinfectants with antibiotics, but it's the same in the clinic as well, where there'd be a high use of disinfectants and biocides within hospitals, potentially to try and eradicate MSRA or C. difficile. There've been big cleaning regimes actually going on at the same time you're kind of getting antibiotic use. So there would have been that co-evolution together, but it's knowing where to put some of the interventions in. So if that selection is occurring at the farm and you need that co-use, can you actually get those mitigation strategies at that farm level, or if it's in the hospital, at that hospital level, before you start mixing a lot of wastes and slurries, be it human waste or animal wastes, together and actually disseminate it much further. So I think it's understanding where that co-selection kind of originates from and actually trying to do the risk mitigation much closer to the source rather than actually once it's out in the environment or at the end of the pipe just about to enter the environment.
Schmidt-Jansen from the Hammer Center of Environmental Research in Germany. Thanks a lot for this very nice talk. Before the antibiotics enter the ecosystem, they enter probably the wastewater treatment plants. And I could imagine that this is a hotspot of exchange of resistance factors. Are there any results or even strategies already how we can handle the problem on the wastewater treatment plant side?
Yeah. It's quite strange that each antibiotic or class of antibiotic will have probably a different temporal or different spatial scale where resistance selection will probably take place. So things like the beta-lactams, which are pretty labile, pretty biodegradable, most of the selection, say from human use, might start actually within the human gut and intestine and be in the sewer prior to sewage treatment. For things like fluoroquinolones, which are slightly more persistent, that resistance selection may extend over a larger kind of spatial scale. So where you would put some of those interventions and measures in would be very, very different. But I know there's quite a bit of tracking. There's a lot of work I know within the UK going on studying kind of selection within the human microbiome. There's a lot of work that potentially the UK water industry might start looking at in terms of different communities and different inputs actually into a wastewater treatment plant in terms of where the resistance hotspots are, because again it's a lot easier to treat maybe a hospital waste going in, a clinical waste going in, or something coming from maybe a particular community where there's a high level of travel going in before kind of primary settlement and secondary kind of treatment than actually end of pipe before it goes in the river. But I think it's somewhere we're starting to see a lot more surveillance actually going in to understand the hotspots and where those potential kind of intervention points may be. But I think the knowledge is coming, but it's not quite there yet.
Well, Professor Snape, really thank you very much for your excellent lecture. A little present from SETAC just to acknowledge your excellent contribution. And thank you so much for your lecture again. Thank you.
Thank you.